All right, so now we move on from kind of our central kind of thought process with oxidative metabolism. We've done glycolysis, we've done glycogen metabolism, we've done the citric acid cycle, and then electron transport and oxidative phosphorylation. And why I like including this next chapter in it is particularly because we can see parallels between electron transport and oxidative phosphorylation in mitochondria and then the processes that happen in chloroplasts. So that's why this is kind of lumped in there. But it's a really cool chapter, and we'll see a lot of homology, and that'll also help us study stuff from our electron transport and oxidative phosphorylation chapter. So kind of some neat history to think about. We'll talk about it a little bit in class, but the idea is Joseph Priestley kind of started doing these experiments when he was looking at the gas given off by fermenting beer, right? We know that to be carbon dioxide from alcoholic fermentation. Noticed that it would put a candle out. But then again, if he did that same experiment and he put a plant in there, that air could be restored. And we know that now because of the process of carbon fixation that occurs during the dark reactions of the photosynthetic processes. But again, the generation of that oxygen allows for that candle to be relit and then burned. So kind of some neat chemistry. But one of the things that we'll spend some time doing is comparing and contrasting the equation that we know happens from fuel oxidation. Fuel plus oxygen gives us carbon dioxide, water, and energy. And then the flip of that, which basically is taking carbon dioxide, water, and energy and then generating oxygen and generating fuel, specifically generating carbohydrates. So we'll spend a good bit of time kind of looking at that comparison, and then also doing a lot of comparison between mitochondria and chloroplasts. So again, really neat process. This process of carbon fixation basically takes carbon dioxide and stores it in a usable energetic form for plants. So lots and lots of energy is stored in this way. As we'll learn, the process of photosynthesis occurs in two stages. We've got the light reactions, and they call them the dark reactions. One of the things we'll learn about as we study regulation of photosynthesis, both of these reactions occur during daytime hours. It's important actually that they not occur while it's dark out, and we'll talk about why that is. But they've historically called them light and dark because light is required for the light reactions, and you don't need light to actually participate in the dark reactions. They often call them light independent reactions, but historically they're called light and dark. But want to make sure to highlight right now, none of the processes that we're going to learn about right now happen when it's dark out, when the sun's not out. As long as the sun's out, we're doing both the light and the dark reactions. Kind of to summarize what happens in these two stages of photosynthesis. Stage one, again, this is these light reactions. We basically are going to do processes similar to electron transport, but rather than the energy coming from electrons that are chemically high in energy, like we get with NADH, what we're going to have is electrons that we make high in energy by using photons to excite them. But other than that, we're going to see a lot of homology between electron transport in mitochondria and electron transport in chloroplasts. A main difference is how we get those electrons to be high energy, by virtue of their kind of standard reduction potential in mitochondria and by virtue of using photons to excite them in chloroplasts. So basically, we're going to be doing electron transport in the light reactions, and we generate a lot of the same species that we do in mitochondrial electron transport. We're going to generate ATP, and that's important because we're going to then use those species, ATP, and we'll talk about, we actually generate NADPH. We'll talk about how that is generated. Again, that's different from something we see in mitochondria, but we'll use these two energy species, ATP and NADPH, that are produced in the light reactions. We'll use those to do reductive biosynthesis in what's called the Calvin cycle. And again, we're going to see homology to chemistry we saw in the pentose phosphate pathway. So it'll be a great opportunity to review that as well, because a lot of the same chemistry that happened in the pentose phosphate pathway, we're going to see happening in the reverse sense. Remember in the pentose phosphate pathway, we actually liberated CO2 and made NADPH. So in this process, in the Calvin cycle, we'll actually use NADPH and use or assimilate CO2 to make carbohydrates. So that's the big picture of the processes that we're going to cover. We'll take about 4 lectures here to go through the entirety of this process. Like we did with electron transport and oxidative phosphorylation, we'll talk first kind of about what the chloroplast is and how we think about energy being used in that. Then we'll actually go through the processes of the light reactions in one lecture, the dark reactions in another reaction or lecture, and then we'll do regulation of the pathway in our last lecture. So we have some neat videos again to watch that kind of help summarize that. Again, we're going to see things very similar to what we saw in electron transport in mitochondria, except again we're going to have 2 photosynthetic reaction centers that actually excite electrons to make them high in energy to pump a proton gradient. We'll see a complex here that is very homologous to complex 3. So we'll see a Q cycle again in chloroplasts. But again, what's different is the final electron acceptor. So again, we're going to see those electrons go to NADP plus to generate NADPH. And we actually get the electrons for these processes, even though they're excited by light, we actually get these electrons by what's called the splitting of water or the oxidation of water. Oxidizing water again generates O2. So again, one of the things we'll do in class in our activity is compare and contrast those 2 kind of simple equations, and we'll put things together so we see how they're connected. So a lot of homology and in some sense reversal of some things that we saw with our oxidative metabolism. Dark reactions, again often called light-independent reactions. We're going to use the NADPH and ATP that we made in the light reactions, and we're going to use that to fix CO2. And so we'd arguably say that this enzyme right here that's called Rubisco is the world's most important enzyme, because if our world could not take carbon dioxide and make it back into fuel for us, we couldn't exist. So we'll spend a good bit of time going through the anatomy of the chloroplast, much like we did the anatomy of the mitochondrion. So we're going to take and dissect this picture, but we'll also compare it to what we saw with the mitochondrion. So I'm not going to spend too much time with this. We'll have a neat activity that allows us to compare and contrast those. So again, these are just 2 review slides from our previous chapter on mitochondrion. What we'll spend a good bit of time talking about that's new is how we use light energy. So we'll have to think about what's present in the visible spectrum of light, right? And we can see that there's lots of pigments that are present in plants, and why there's a whole range of pigments is they each have lambda maxes that are different. So they're each able to kind of help and gather light energy from a different region of the visible spectrum. We'll talk a little bit about some of the parts that are within these light-absorbing pigments, so the chlorophyll molecules, and we'll compare and contrast what we see, for example, in the cytochromes in mitochondria, where we see hemes, so iron-containing protoporphyrin rings, and then the hemes, or the modified hemes that we see in chloroplasts, which don't have iron, they have magnesium, and again some different functionality, including a 5th ring here and so forth. So we'll spend a little bit of time just comparing and contrasting those. Again, this concept, I'm going to flip back here just for a second. When we have all of these different pigments, we normally think of chlorophylls being green, and that's these chlorophylls A and B. But we have all of these other pigments that we call accessory pigments. And so those allow us to just look at the entire spectrum of visible light and use all of it to our advantage. We'll do a little activity in class that helps us think about how chloroplasts harvest light. And the idea is that you have a whole bunch of these chlorophylls that are what we call antenna chlorophylls. They don't actually do photosynthesis there. They're just responsible for kind of harnessing light. So that's these light green ones here. These dark green ones are actually the photosynthetic reaction centers. That's where the chemistry happens. But the bulk of the chlorophyll that's actually within chloroplasts is just for harvesting light. And so we'll talk about what happens when you can absorb light. This is just a picture to show those light harvesting complexes. Here you can see all of these different chlorophyll molecules here, where we have these heme groups with the magnesium at the center. But if you remember, if you've learned anything about spectroscopy, it's all about matching energies, right? You've got gaps essentially between levels that electrons can kind of travel. And when you match that energy band, that energy gap, you can absorb energy and kind of jump to a different level. So again, when light gets absorbed, and I'm not going to spend a lot of time talking about kind of the spectroscopic part of this, but when we absorb light, there's lots of things that you can do as that energy is, again, released back out. You can have what we call internal conversion, you can have fluorescence, you can have photooxidation reactions, but the important one that we'll talk about two that we'll talk about are photooxidation, which has to do with the loss of electrons, and then what we call exciton transfer or resonance energy transfer. So again, we'll talk about what some of these other processes are, but the two main ones that we'll talk about are exciton transfer and then photooxidation. Because the idea is, I'm going to flip back here for a second, if you are an antenna chlorophyll, you are going to have your electron get excited, but you're not going to lose your electron. Your electron doesn't become part of the electron transport chain. Rather, you're going to transfer your energy to an adjacent chlorophyll. And that's going to happen. That's what these little white lines represent, is the resonance transfer energy being transferred between all of these different antenna chlorophylls. When you finally get to one of these dark green ones, these photosynthetic reaction centers, once that one gets excited, then we have photooxidation, and that electron becomes part of the electron transport chain. So again, we'll talk about that in some detail in an activity, and we'll actually kind of act out and role play what happens in a chloroplast. So again, what we'll have is electrons that get excited in these reaction center chlorophylls, and they'll go through the electron transport chain here. But again, those electrons need to be replaced from something, and so they get replaced when we do what's called the oxygen splitting or the oxygen evolving center. We take molecular water, we oxidize it, those electrons go back and replace the chlorophyll electrons that were oxidized, and that's how we generate oxygen. So we won't get into the details of how this electron transport process occurs until our next lecture. This lecture was just introducing the chloroplast and, again, some different chemistry that happens when we consider using photons of light in part of the process.